So in the last lecture we met optogenetics, which is the use of transgenic technology to enable us to gain really fine-grained control of neurons. And what I want to do in this lecture is to talk you through a classic experiment using optogenetics to try and reactivate an engram. So the ability to selectively manipulate neurons means that we can get really, really fine-grained control, not just of entire regions of the brain, but of selected neurons within the brain that may not be spatially co-localized, so they may be kind of scattered. So this is a really incredibly important tool. And it makes use of promoter technology. So we met promoters in the last couple of lectures. Promoters are segments of DNA that control genes, and if we can control the promoters, then we can get control of the genes ourselves. So a promoter that's been important for manipulating the engram is the promoter for a gene called CFOS. So CFOS is an immediate early gene, which is one of a class of genes that get activated when something happens to a cell. So if it suddenly becomes active for some reason, CFOS switches on. What you can see here is an example of neurons in the dentate gyrus. So you can see the two blades of the dentate gyrus, and the red spots are neurons in which CFOS has been activated by some event. And in this case, the CFOS promoter was used to also cause the cell to express a reporter protein. So the reporter is what's making the cells glow red in response to fluorescent light. So we can see which cells are expressing CFOS by which cells are expressing this reporter. So by linking genes to the CFOS promoter, you can get control of cells that had been active. So this technology has led to this very cool experiment. The idea is to try and get control of the neurons that were activated by an experience, and then to reactivate those neurons experimentally and make the animal have that experience again. So that was the basic idea. It's an amazing sort of science fiction idea. So the first step is to create a new memory and to activate the immediate early genes, including CFOS. And then the second step is to use that CFOS promoter to insert opsin genes only into those CFOS expressing cells. So now we'll have channel rhodopsin only in the cells that were activated by that experience, and that means that we can get control selectively of those neurons. So just to remind you of what a promoter looks like, so this is the structure of the gene, and the gene, in this case the CFOS gene, is shown by the red in the middle there, and the promoter is the part at the start. And the promoter means that we can tag the neurons that were involved in the whole sequence of switching on the CFOS gene. Now in this experiment they also made use of another promoter called TTA. So the TTA gene is a gene that makes a TTA protein, and it's sensitive to antibiotics such as doxycycline or tetracycline. So in this experiment they made use of that gene to control the channel rhodopsin. So the construct is shown here. So you can see the gene for the channel rhodopsin, that's the CHR2. It's linked to a reporter gene for yellow fluorescent protein, so that's the EYFP next to the channel rhodopsin. And those two genes are under the control of this regulatory promoter sequence at the start. And though that regulatory sequence is controlled by the TTA gene. So the protein made by that gene, the TTA protein, will cause channel rhodopsin to be expressed, and also the fluorescent protein to be expressed. So that means we can see which cells that happened in. So CFOS activation switches on TTA, and that switches on channel rhodopsin. So that's the basic idea. We're daisy chaining together these sequences of promoters. So then the final ingredient in this experiment that made it possible to get really fine-grained temporal control of all of this is the fact that the TTA gene can be controlled by antibiotics. So if the doxycycline or tetracycline or whatever is present, that gene will be switched off. If it's not present, then the gene will be making TTA protein. So normally the TTA protein will bind to the promoter and switch on the channel rhodopsin gene, for example. But if we give the animal doxycycline, then that won't happen. So that means we can control whether or not the animal is making channel rhodopsin in any of its cells. So this is how the full experiment unfolded. So the first thing was to take the channel rhodopsin gene construct, so that's the channel rhodopsin and the yellow fluorescent protein with the TTA promoter in front of it. So package that all up in a virus and inject it into the dentate gyrus. And also, at the same time, give the animal doxycycline so that none of that was going to be activated, because you remember that the doxycycline suppresses all of that. So the animal's got these genes in cells in its dentate gyrus, but they're not doing anything as long as it's having doxycycline. The second thing was to take the mouse that's got the doxycycline and to put it into a context, the pretty benign context, a box, and let the mouse experience that box and get used to it and activate place cells and activate CFOS in those cells and all of that kind of stuff. So learn about that environment and learn that nothing particularly bad happens in that environment. Then the mouse was taken out of that box and it was placed in a different environment and taken off the doxycycline. So now the cells have the capacity to make channel rhodopsin in the cells that have the CFOS promoter active. And that will be the cells that were in which CFOS was switched on. So what they do now is switch on CFOS by giving the animal an unpleasant experience. So they give it a foot shock. The foot shock activates CFOS in a whole bunch of cells. All of those cells start making channel rhodopsin. So now you've got an animal that has channel rhodopsin in the cells that were activated by the unpleasant foot shock experience. Then the final step in the sequence was to take the animal, place it back in the first context, back on the doxycycline, so no more channel rhodopsin is being made, and now shine a light into it. And what so down an optic fibre and into the dentate gyrus. And what that light will do is it will activate channel rhodopsin in the cells that had been activated by the foot shock experience. Now the mouse had never had a foot shock in this benign context, but in the mice where the light was shone, those mice froze in terror as if they were recalling the bad experience. And this is shown here. So you can see on the left is the data from the mouse with the full gene construct, and in the right is some control data from mice that had the reporter gene, but they didn't have the channel rhodopsin gene. So everything else was the same, but they didn't have the channel rhodopsin. And you can see in the mice with the channel rhodopsin, those mice froze. So the implication is that the shining of the light had reactivated the engram for that foot shock experience, even though the mouse was in a new environment that hadn't had the foot shock in. So it was reminded of the foot shock. So this is really strong evidence that the engram for an experience like a foot shock is in the hippocampus, and that we can reactivate it and make the animal remember that experience. Now, of course, we can't ask the mouse exactly what it does remember, but the fact that it freezes in fear suggests that it has remembered something about that experience. So this was a really landmark experiment, and follow-up experiments have done things like alter the engram post hoc, so take a unpleasant memory and convert it into a pleasant memory and so on. So these methodologies have given neuroscientists the ability to not just visualize the engram but also to manipulate it, which is really an amazing thing. And it takes us right back to what we began this section of the course with, that trying to find the location of the engram in the brain, and it now looks like we're getting closer to answering that question, although we still have a long way to go. So in the next set of lectures, we're going to be looking at memory and how it unfolds across the lifespan, particularly in development and also in aging.